Hi, my name is Andrew Piaget, and this is my research, which has to do with investigating the occurrence of resistance in 3D7 Plasmodium falciparum parasites, which was performed in Ghana, West Africa. So malaria, as we researched, is caused by the protist parasite Plasmodium falciparum, which is one of the major causes of death in tropical regions of the world, having sub-Saharan Africa being affected the most by this phenomenon or by this disease. So for this research, the hypothesis was to use subtherapeutic doses of an anti-malarial called their artesunate to produce resistant lab strain 3D7 Plasmodium falciparum in vitro. Um, so the tests done for this particular research where we first cultured 3D7 lab strain Plasmodium falciparum, determined the infected to uninfected red blood cells in media collected. Secondly, we determined the inhibitory concentration of the drug at 50%, um, at 50% of parasites, which is known as the IC50. Thirdly, the parasites were recultured in 10, 20, and 30, 10, 20, and 50 dilutions of the artesunate drug over 12 days. As shown in figure 3, that's our method process. Figure 4 shows our IC50, and figure 5 and 6 are the results. So after we did this, we focused on a gene called the K13 gene, which is one of the genes responsible for resistance in Plasmodium falciparum. After these tests, we extracted the DNA of our cultured 3D7 Plasmodium falciparum parasites, and sequenced their data and analyzed their data to determine if there are any mutations in that resistant gene mentioned before, K13. So according to our conclusions, the results show that the treatment of 3D7 Plasmodium falciparum parasites with subtherapeutic doses of our anti-malarial artesunate did not show or did not lead to any mutations in the K13 propeller gene. This shows us that there might be longer periods of cultivation needed to be able to see these mutations, which have been seen in other texts as well for this particular research. I would like to take some time to extend my thanks to Dr. Kristen Picardo for her never wavering support for my research and research success at St. John's Fisher College. I'd also like to extend my gratitude to Dr. Janelle Maticio, who guided me through my research as an advisor and was a pillar of my success in this particular research as well at St. John's Fisher College. I'd also like to extend my gratitude to Dr. Michael Ofori, who served as a research advisor at Noguchi Memorial Institute of Medical Research back in Ghana, West Africa, and this institute was under the CDC and WHO. So I'd like to extend gratitude and thanks to them as well. Thank you, and thank you for the opportunity to present.